Today I will be presenting our needs-driven and evidence-based approach to point-of-care technology development. This presentation is brought to you by the UC Davis Lawrence Livermore National Laboratory Point-of-Care Technology Center. We are an NIH-funded site. I am Daniel Mecozzi, a junior specialist at this center. Our center theme is point-of-care pathogen detection for emergency and disaster care. Now I'll present how needs assessment affects our cores, beginning with Core 1. The goal of our needs assessment program is to define end-user needs. To accomplish this, we used an online survey tool, SurveyMonkey, to survey the readership of the point-of-care journal. Survey questions include visual logistics for easy-to-comprehend questions, multiple-choice questions analyzed by Pearson's chi-square exact tests, ranking questions analyzed by ANOVA and Tukey's multiple comparison tests, and trade-off responses analyzed by multivariate logistic regression. This graphic displays two different sampling methods. On the left is a coupled direct method using an adapter, and on the right is a direct method using a built-in retractable needle. All of the questions in this presentation were taken directly from our survey. This graph shows the result for the previous question. The white bars represent the direct method, while the black bars represent the coupled direct method. The x-axis is setting, and the y-axis is frequency of selection. Looking at the left bar, U.S. and foreign respondents preferred a direct sampling method in a disaster setting. This graphic displays two different sample processing methods. On the top is a device that processes multiple patients for a single pathogen. On the bottom is a device that processes a single patient for multiple pathogens. This graph shows the results for the previous question. The white bars represent the multiple pathogen single patient method, and the black bars represent the multiple patient single pathogen method. The x-axis is setting and the y-axis is frequency of response. Looking at the middle and right bar, U.S. and foreign respondents significantly prefer multiple pathogen testing in urgent care and emergency room settings. This graphic focuses on biohazard disposal method. On the top is a device with a waste storage reservoir. On the bottom is a device that stores waste in the disposable test cassette. This graph displays the results for the previous question. The white bars correspond to the disposable test cassette, and the black bars correspond to the waste storage reservoir. The x-axis is setting, while the y-axis is frequency of response. U.S. and foreign respondents significantly prefer a self-contained disposable test cassette method of waste disposal storage in the three settings we surveyed. To summarize the impact of needs assessment on Core 1, respondents prefer a direct sampling method in a disaster setting. Respondents prefer single sample, multi-pathogen analytical processing in emergency room and urgent care settings. Ideally, the device should use a disposable test cassette that doubles as biohazard waste disposal. Also, the device should be highly sensitive and portable. Moving on to Core 2, I will show you how important needs assessment was for designing our RFA, novel H1N1 influenza multiplex testing at the point of need. Here is the actual question from our survey. Respondents were asked to design and rank a pathogen test cluster. If you would like to participate, please email me your response. This graph displays the results of the previous question. The x-axis is the pathogens, the y-axis is the weighted score. We found that novel H1N1 influenza was significantly preferred for pandemic respiratory testing by U.S. respondents. Additionally, U.S. respondents felt that it was necessary to equip disaster response teams with pandemic testing to include novel H1N1 influenza, avian influenza, and severe acute respiratory syndrome. In summary of the Core 2 program, our needs assessment identifies gaps in point-of-care device design and couples technology development with end-user defined design criteria. Our needs assessment also identified novel H1N1 influenza and avian influenza for pandemic respiratory pathogen testing. Moving on to Cores 3 and 4, I would like to focus on the effect of needs assessment on our challenge grant, highly multiplexed late PCR point-of-care pathogen detection for critical care. Here is the actual question from our survey. Respondents were asked to rank pathogens from 1 to 10 to create an ideal test panel for a disaster setting. This graph shows the result from the previous question. Respondents significantly preferred testing for MRSA and Vibrio cholera at the point of care in a disaster setting. Our RC1 Challenge Grant uses late PCR to detect multiple key pathogens, including MRSA. The point-of-care late PCR assay will impact patient care by facilitating early, directed antimicrobial therapy. On the left of this slide is the prototype late PCR device already in existence. The organisms identified here show the multiplexing capabilities identified in the grant. Our center believes needs assessment is so important because it drives technology development in an evidence-based fashion. We would like to propose some concluding recommendations. Number 1. Needs assessors need to link device designs, test clusters, and points of service. Needs fulfillment has been fundamental to point-of-care technology development throughout its modern history. Evidence-based needs confirmed statistically drive current projects locally, nationally, and globally. As an example, our 2009 RFA derives from evidence-based point-of-care needs assessment, linking cores 2 with cores 3 and 4, and implementing strategic concepts inherent in the original design of the center. Point-of-care novel H1N1 influenza and multiplex testing will address a critical national problem at the point of patient and caregiver need, thereby closing the loop. High mortality for undiagnosed acute coronary syndrome led to evidence-based surveys to determine strategic plans for rescue of myocardial infarct patients. Optimal point-of-care cardiac biomarkers such as the new Cobas H232 handheld provide a sustainable infrastructure in which manufacturing can meet clinical demand and integrate into small world networks for managing, diagnosing, and treating in rural underserved areas lacking adequate personnel. Number 2, leaders need to provide the roadmap. The international trend is toward cost-effectiveness studies or at least comparative effectiveness analyses for policy recommendations. The National Institute for Health and Clinical Excellence is working in the UK and 40 other countries to align healthcare coverage and comparative effectiveness of drugs, devices, and delivery systems. The United States lags in this fundamentally important effort to direct appropriate funding based on objective assessment of needs and outcomes. The NIBIB is providing badly needed leadership and exemplary Core 3-4 roadmaps in needs assessment that will help drive rational development of point-of-care technologies. Demographic trends and surveys identified that a diabetes endemic has stricken Thailand. Setting up small world networks and rolling out point-of-care for hemoglobin A1C, per recommendation of the American Diabetes Association International Expert Committee, provides a sustainable infrastructure for diagnosing, monitoring, and treating, potentially mitigating costly adverse side effects and improving patient outcome. And 3, integrators need to create sustainability. Needs assessment, expertise, and knowledge should be shared, disseminated, and codified by the NIBIB Point-of-Care Technologies Research Network for Sustainability, just like the notion of point-of-care testing was codified in the 1990s. Financial sustainability by nonprofits is threatened by recent economic setbacks, but fast growth in many emerging markets appeals to industry as Western markets stagnate and point-of-care testing is expanding rapidly worldwide. Therefore, the NIBIB Center's model can move needs-based and evidence-based point-of-care technologies, that is, ones deemed to bring sustainable effectiveness and value, not just low cost, to the doorstep of everyday implementation in a variety of settings, such as critical emergency disaster care, public-private partnership, and low-resource countries. Here is a list of our references. This presentation was supported by the National Institute of Biomedical Imaging and Bioengineering Point-of-Care Technologies Grant. This content is solely the responsibility of the authors and does not necessarily represent the official view of the NIBIB or NIH. That concludes my presentation. Thank you very much for listening in.